So let me start my start the presentation. Is it visible?
Yes, sir.
Yes, sir.
Thank you. So I think in my last lecture, so all of you were there, na? In my last lecture.
Yes, sir.
Okay.
Yes, sir.
I think you have got this slide. I send it to sir to circulate among you. Do you have this one? Hello. Do you have this slide with you?
Yes, yes, I
Yes,
have
sir.
shared it.
Okay. So it will be easier. In few sentences, I will just explain what I said in my last lecture. Okay. Suppose, I hope you all remember. If you don't, please interrupt me and do not hesitate and ask me if something is not clear to you. Okay. So this is the barrier, physical barrier for a pathogen to enter inside the body. Somehow the pathogen manages to get inside. So what will happen? There are some cells. In the last lecture, I mentioned about two cells, basically dendritic cells, macrophage, and I also mentioned there another cell, that's the B cell. These are called the antigen presenting cells or APCs. What they do, when this pathogen enters, this cell will phagocytose them, and then they will chop it into pieces, and through MHC2 molecule it will expose it to another T cell called T naive cell or T0 cells. Not only expose the to T cells through MHC2, it will also release some interleukins like IL-12. And there are many other. I am just mentioning here the main one, the IL-12 for you. Then what happens? These T0 cells, these are, you must know two things. These, just remember it right now, these cells are CD4 positive. What positive? CD4. Here as I have written CD8, but these cells are CD4 positive. What does that mean? It means these cells, these T naive cells or these cells, on their surface they have some protein molecules. They are called cluster of differentiation 8 or 4. That's why they are called CD4. And as they are having these proteins on their surface, they are called CD4 positive because they are having it. They are not having CD8 on their surface. So they will be CD8 negative. Am I clear to you so far? I'm clear to you. I'm just repeating the last lecture. Am I clear to you, students? Just say yes or no. Someone.
Yes, sir.
Okay.
Sir.
So what will happen then? These T naive cell to T helper cell 1. And what this T helper cell will do? It will again activate the innate immune system, like macrophage itself. It will make a super active macrophage, or epitheliocyte cells, then they will fuse. There are many types. So these Th cells secretes, there are many things they secrete, among them interferon gamma, TNF, these are also secreted and they activate the innate immunity. Clear so far? Now, these T helper cells, Th1, they secrete some interleukins. Those interleukins 2, here it was 0, then it is 1 and it is 2. Okay, and here interleukin 12. You can, I don't know whether it will be very feasible, but you can try to remember this way: the interleukin 12, then T0 will be TH1, and this will release IL2. Just to remember it, a way of remembering. That's what I said. So interleukin 2 will be secreted by TH1. These Th2 interleukin 2 has two functions. Either it acts on itself, that is autocrine function, or it will act on other T cell, that is CD4 8 positive T cells. Okay, so these, don't confuse that these Th cell will get converted into T cytotoxic cells. No, this is a different cell. It is secreting IL2 and it is acting upon these cytotoxic cells, T cytotoxic T cells, or CD8 positive cells. Okay, what I mentioned in my last lecture, I think you remember that these antigen presenting cells or APCs, they have what they have on their surface, MHC2 molecule. Then this MHC2, then the whole thing is happening. One, second, there is all the nucleated cells, they have what I have said, they have MHC1, right? I think I mentioned it in my last lecture. So if a pathogen hides itself, suppose this pathogen, upon entering the body, not getting exposed to dendritic cells, macrophage or B cells, they are, what they are happening? They are hiding themselves inside a cell of, suppose, a normal body cell. So what will happen? So that cell will be affected. So how will you detect that? If those, sometimes what happens, those cells kill the pathogen inside and they expose it, those uh, some fragments of those pathogen outside through MHC1 molecule. Okay, and these cytotoxic T cells recognize such cells and they kill the affected cells through some molecules. Here I have written the name of two things, that perforin and granzyme. So what they do, this perforin and granzyme, these T cytotoxic cells, it is like a very, how I should say, some kind of, just imagine a chemical being released and getting, and upon, after being released, it makes pore on the affected cells. Here the infected cells, it is its target one. So these granzyme and perforin will make pore on the, say, on the surface of the affected cell, and it will, that will lead to the death of that very infected cell. So the infection will not spread. Is it okay so far? Is it okay so far? Okay, thank you.
Yes, sir.
So this is a pathway one. Then another one, and the third one I mentioned was IL4 signaling. secreted by a unknown cell. There are nowadays there are some cells are also mentioned who secretes IL-4. I have not mentioned it here. So what happens? These cells also affect on CD4 positive T naive cells. What will happen then? These lead to that uh, these naive cells will change into T helper cell 2. Okay, as this one was secreting IL-2, interleukin 2, these will secrete IL-4 and 5. These will secrete IL-4 and IL-5. These are the very important. I have written the most important one. Okay, there are many other things you will find. If you go when you will study MSc, you will find there are many other molecules. Many things are also there. I am just talking about the important one for the examination purpose or the basic one. So these IL-4 and IL-5 will affect all cell lymphocyte. That is also now I'm coming to that another cell called B cell. What is that name? B cell. That is also a when it is affecting on that B cell, that B cell was also naive. It doesn't know anything. Just before exposing to getting exposed to IL-12, IL-4, it had no idea what is its function. So it could be lead to this way or this way as well. Similarly, there is another naive cell called B cell, not T, that is B cell. When IL-4, IL-5 act on those B cells that get activated, then it will become activated B cells, or here it is, then it will secrete antibody, or the cells are activated B cells, we can call these as plasma cells. And for your information, these plasma cells produce, as I said, antibodies, live for few days and can produce antibodies at a rate of around 2000 antibodies per second. Just imagine the amount of protein it is producing per second with high precision only. We all know antibodies are very specific, so it has to be specific and at a rate of 2000 antibodies per second by a cell. So the how active the whole protein manufacture machineries are can be easily imagined from this number of 2000 antibodies per second. Okay, so far it was okay. So let me come over here. I have already mentioned few cells: T cells, B cells, T helper cells, CD4, CD8, and plasma cells. Right now I mentioned. So these are all, after these are all lymphoid progenitors. These are all originated from lymphoid progenitors. They are called lymphoid cells. Okay. So there are three types of lymphoid cells: T cell, B cell, and NK cell. NK cell or natural killer cells. As I have mentioned, so far I have mentioned that T cell, B cell are part of adaptive immunity or acquired immunity, whereas the other things which are the cells, the dendritic cells, the macrophage, these are all part of innate cell. There is one cell which is which has the origin or which can be defined as lymphoid cell, but it is a part of innate cell. What is this? NK cell or natural killer cells. So what is the speciality of natural killer cells? As the name suggests, as the name suggests, it's a killer machine. It's a killing machine. Okay. Generally, what happens in my previous slide, suppose you land upon the inside the body, and now you need someone who will have no mercy on cells which are affected or which are having, mm, what I should say, this pathogen inside. So, first, what will be the first response? Just to kill the pathogen, okay, by any means. For those cells, we have natural killer cells, NK cells. Do you get my point? Hello? So
Yes,
far,
sir.
am I clear to you, or you want me to repeat it, what I am saying?
No, sir. It's clear.
Okay. So there is a cell, NK cells. It is the cell. Uh, it has large granules, just like the perforin, the granzyme, which I have already mentioned before. It is also having perforin granzyme, and it plays and it kills those cells having the pathogens. Now, one thing you must know, that these cells exhibit spontaneous killer activity against any foreign cells, tumor cells, pathogen infested autocells, etc., even in absence of prior stimulation. So it doesn't, this cell does not require any previous experience. It can kill, again I am mentioning it, why these cells are so important, because these cells can kill any foreign cell. It can kill tumor cells. It can kill pathogen infested autocells. As I mentioned, the cell may remain hidden inside a self cell, so it can also be killed by uh, NK cells. Okay, so these cells provide the first line of defense for some infection or tumor growth, or if there is any pathogenic alteration in the tissue homeostasis. Means if their pathogens are affecting self cell, I mean the body cells, it can also be killed through NK cells. So NK cells are not phagocytic. So what I have said, there are macrophages, dendritics, what I have said already before, that macrophages, dendritic cells, these are phagocytic cells, but NK cells are not phagocytic cells. Clear? Am I clear to you, all of you?
Yes, sir.
Okay. So I have mentioned what I have mentioned so far. The NK cells, T cells, B cells, these are all lymphocytes. And what are the other type of cells where they are getting formed? These are all happening inside our bone marrow. From where these cells originate? They originate from some stem cells. There are other variations. I'm not going into that, but you must remember from hematopoietic stem cells, all these blood cells originate. Whether these T cells, B cells, or macrophage, dendritic cell, mast cell, eosinophil, you have all heard neutrophil, they all originate from where? From hematopoietic. They have the lineage of hematopoietic stem cells. One One among them is lymphoid, another one is myeloid. Here in this picture, one blood cell is missing, that is RBC. RBC is also a myeloid, is originated from the myeloid progenitor cells. So these are the part of innate cell, innate immunity, and these are part of adaptive immunity. Now, there are few things or that requires. Okay, before that, okay, one thing I should mention here. As I said, all these cells originate in the bone marrow, right? So why T cell? Do you have any idea? I think you might have some idea why it is called a T lymphocyte. Anyone? T? Why it is T? Anyone? Any idea? Okay, no idea. So this T stands for thymus. Cells from bone marrow goes into or into the thymus where it gets matured. I will come later on, and then they come back into the circulation or in the blood, in the peripheral circulation. We call uh, the where it will be either CD4 positive or CD8 positive. Okay. Before that, I sometimes it comes into question what is the basic difference. How you will recognize these three are lymphoid cells. What are the basic difference? These cells, if it is a B cell, it is having CD protein, which one? 19, CD19 and CD20. If this is a B cell, it will be having CD19, CD20. Whereas if it is a T lymphocyte, it will be having CD3, THR 3. So this is 1920, 1920. This is be having on the surface some protein called CD3. It will be having some protein called CD19 and 20. And these cells, NK cells, they are not having either of either CD3 or 19 or 20. So it is little bit different and we call them NK cells. Now, I don't think you require that much because you need to know more because there is another division of null cell. From null cell in comes NK cell or killer cells, that is antigenic dependent. There are many other divisions are there. I don't think you need to know all this at this moment. Okay. So if you remember this, I think that will be the basics which is required for you. Okay. So two cells I have mentioned. The most important two cells are B cell and T cell. So B cell and T cells, they are having some receptors on their body. It is also there in your syllabus. So T cell and B cell cells are having some receptors on their surface. So what do we call the T's are the receptor. This is a receptor present on the B cell. And what do we call it? Any idea? I think you should know. What is this? Anyone from the
Sir
structure?
wants to know what is the receptor of the B cell.
Just the
I think
figure.
okay.
This, anyways,
Sir, antibody.
antibodies or immunoglobulin. Okay. Okay. Very good. So
Good, Rian,
immunoglobulin.
good, very
Good.
good.
Or
I think your seniors should have come up with the answer. Very good.
so this is immunoglobulin or antibodies. Now, if you compare with these cells, these receptors, just see what is happening. is the basic difference? There are only two chains, peptide chains, alpha and beta. Here they have a small one called light chain and heavy chain. Again a heavy chain and a light chain. Okay, structurally this is the difference. So here it has four chains, here it is having only two chains. Alpha beta, here it is having light chain, heavy chain, again heavy chain and light chain. And this area where the antigen binds with the antibody. Antigen binds with the antibody. And here also they bind, antigens bind. So if we consider one molecule, we will find one antibody molecule is able to bind with two antigens, and one T cell receptor is able to bind with only one antigen. Okay, these immunoglobulin or antibodies may remain on the surface or may be secreted. So comes out of the cell and may roam in the body freely in the. Hello, is there any question? Okay, no, I think so. So these antibodies may remain with the cell surface attached, or may be secreted, and they may roam freely inside your body. But these cells, these antibodies are themselves the T cell plasmid only. The basic. So you will face two words in your examination. Two terms, avidity and affinity. What are those things? See, one molecule is having an option of binding two antigen molecules. Suppose this one gets secreted and two of such molecule, they are uh, they are binding, they are making two molecules are getting attached together, getting secreted from the cell surface, and two molecules are getting attached with each other. So how many antigens they can bind? Four, right? Here it will be only one. One molecule of B cell and one molecule of T cell. So you will find has the ability to bind two antigens, and it has the ability to bind one antigen. So if I rearrange these regions, there will be simultaneous binding of two identical antigens, thereby increasing the total strength or avidity of the interaction. What does it mean? Means the antigen can bind here with this molecule in two places, or two antigens can bind here. Okay, only one is possible here. So it has more probability to bind with uh, more strength in comparison to the areas or the number. So it is called avidity. Okay, so this is the avidity. And what is affinity? Affinity is, suppose this one and this one, you don't have to bring this into consideration. Only one, how strongly an antigen will bind with this portion, or how strongly the antigen will bind with this receptor, how strong it will bind. That is affinity. More the affinity, stronger the bond. Okay, more the affinity, stronger the bond. So sometimes I have seen that question comes, what is avidity and affinity? Okay, now as I have mentioned, this is a antibody molecule. it may remain on the cell plasma membrane surface or may be secreted. Okay. What I have already said here, that if a presenting cell can promote via so many things, can promote the activity of B cells and can start secreting antibodies. Right? Am I clear so far? And those antibodies are B cell receptors, as I mentioned. Now you tell me, suppose you are getting, someone asked me a question last time about memory cells, right? Someone asked me question about memory cells. Here the comes the question, the answer to that question. You have an activated B cell. So what will happen? The B cell will produce antibody and it will destroy the pathogen. Ultimately, it will destroy the pathogen. So far, it is fine. But just imagine one antigen presenting cell has to trap the pathogen, then it will process it, then it will bring it to another cell, which will get converted to another cell, which will again then activate another cell. So much, such a long process. It takes around 5 to 10 days for when the cell on the first time a body is getting infected with an infection or pathogen, it takes 5 to 10 days to produce this much of, to get activated B cells and antibody titer, high antibody titer into your body or into your bloodstream. So it takes time. How much time it takes? Almost 5 to 10 days. But it is quite possible that after some time you may again get affected with that same pathogen. So if you have to go through that very process, then it is time consuming and it can cause harm to you. So to reduce that possibility, cell or the body has created a mechanism. Instead of, after creating activated B cells, it will also create some other cells. They are called memory cells. It will be having the memory, what kind of antibody to be produced by me. The cell will know what kind of antibody to be produced by me if I encounter this very antigen once again. So I am repeating it. If you are getting infected with that very pathogen, then these memory cell will immediately start producing antibodies. So the titer which you the time it took almost 5 to 10 days, here it will take max to max 3 days. Within 1 to 3 days you will get those antibodies in your circulation. So probably you will not realize that you had that infection. Even before you realize, you will find that antibody, that pathogen has been destroyed by those antibodies. Pathogens have been destroyed by those antibodies secreted from memory cells. Okay? Do you get my point? Now tell me if there is anything you are not clear about. Am I clear to all of you?
Please respond. Sir is asking question.
If
Please
I'm
respond, students.
If not clear, please ask question. If cleared, please say
no,
yes or no.
okay. Anyways, okay, fine. So, what I have mentioned, if the infection is new, then it will take 5 to 10 days for the system, body system to produce antibodies. Second, if it is old, if it is again you are getting infected with that very pathogen infection, then it will take only, or it is called the secondary infection. Primary one 5 to 10 days, secondary 1 to 3 days. Second uh, is that mm, the amount of and the specificity, that is very important. In the primary infection means when the first time you get infected with a pathogen, the only an antigen binds with antibody is less in the primary one, but in the secondary infection it is much more tighter, or the affinity is higher, so it is easier for antibodies, or the system I rather, to kill those pathogen. Okay, it recognizes in a much better fashion. I don't know whether you have started enzyme or not. I think probably your sir will teach you. I just, for the sake of your examination, I would like to mention one term that is called dissociation constant. You don't have to remember, just keep it in mind. It will be easier for you. We use dissociation constant for the estimation of affinity, or how strong the binding is. Smaller the number, stronger the binding. It is always smaller the number, stronger the binding. Okay. So in primary infection, it has been found that dissociation constant is around 10 to the power minus 7 per molar. Whereas in the secondary infection, just for your knowledge, okay, if you, it is there in your exam, then if you write it, it will fetch you better numbers. And in the secondary infection, it is 10 to the power less than 10 to the power minus 11. So the number is very small. It means the specificity, the affinity is very, very high in the secondary infection. So such thing you won't find with these T cell receptor as it is there in your syllabus. So these are the things which I have mentioned so far. These are the differences between T cell and B cell receptor. One more difference is there that is very important. As you have said, this is antibody or immunoglobulin can recognize carbohydrate, protein, lipid, DNA, small molecules. Okay, so you name it, you will find all are there. What I said, small molecules, proteins, DNAs, lipids, carbohydrates, you will find all are recognized by these uh, antigens, but you won't find such uh, variety in case of T cells. It is mostly uh, recognizes peptides that is bound with MHC molecule 1 or 2. Why I said so? because T cell recognizes peptides either from MHC2 molecule or MHC1 molecule. So these are the basic difference between T cell and B cells. Okay, receptors, T cell and B cell receptors. I think I have 10-15 minutes more, right?
Yes, sir.
Okay, so within 15 minutes I will finish.
Take
Now,
your time, sir. Take your time, please.
no, no, as I said, within 1 hour because the students will be bored otherwise. Anyways, okay, as I said, this is where these cells develop primarily in the bone marrow. Then B cell will directly enter into the circulation, that is peripheral circulation, the blood circulation, uh, peripheral circulation, whereas T cells will first go into the thymus where they will be selected or trained, then it will come back again, then it will come back into the peripheral circulation. Okay, so what is this, what is what, let me give you an answer, an idea, sorry. Here you can see a B cell. What is this? A B cell. A, from, this has come from where? A stem cells. This is called common lymphoid progenitors. From here, different types of lymphoid cell can be produced. So then in the B cell, in the bone marrow, they will change or differentiate into pro B cells. This is a thing you just that will happen inside the bone marrow, and this is the mature or mature B cells. Okay. Here, what happens? The B cell development, how B cells are developed, B cells activation and cell function. B cell development begins in the bone marrow where hemopoietic stem cells go through the process called hematopoiesis to become common lymphoid progenitor cells. This one. Before differentiating to T or B cells. So what I said, this is the common lymphoid progenitor. From here it will be B cell, it can be T cell, or it can be an NK cell. So for here our consideration is B cell. The earliest B cell known as pro B cell. This is the first one, pro B cell. At this stage of development, the cell do not express any immunoglobulin. What I said, the ultimate aim of a B cell is to produce antibodies. But here you will find any immunoglobulin or antibodies on their surface. But they do express other B cell markers such as CD19. I said for how to recognize a B cell through some markers on their surface as is CD19, CD20. Okay. So it is having CD19. That's why we can recognize this cell as B cell. This is the first one, so we call it pro B cell. So then after pro B cells, they regenerate begins. You don't have to remember it because it will, you will study in your MSc probably. These progenitor cells will go VDG, gene rearrangement. What is that? So I'm not going into this. See, we all know thousands and thousands of pathogens enter our body that get recognized by antibodies. How do they get recognized? For that, you should have, if you have different antibodies for different pathogens, so you have thousands and thousands of genes. Basically, it is said, as anyone, do you have any idea roughly how many genes do we have in our body? How many genes do we have in our body? Any idea? Anyone? No idea. Okay. Roughly, it is believed that we have around 30,000 genes. If you consider alone these antibodies, they can bind to various types of antigens. As I said, they were very high. Uh, it can bind with DNAs, it can bind with small molecules, proteins, lipids, anything. You name it, you peptides, you name it, you will find it can bind. So how that variability? So for that, they have a special system called VDJ gene rearrangement. By rearrangement by or shuffling of some genes, they can create the variability among them. Okay, different types of antibodies. So for that, so, actually, who discovered any? I do you have any idea who discovered it? I the list I gave you today in previous lecture. Here you will find in 1987 Susumu Tonegawa. Susumu Tonegawa, he actually described this process of gene rearrangement for antibody production, various different types of antibody production, and he got Nobel Prize in 1987 in in physiology and medicine. Okay, so you don't have to remember that. What is VDJ or so in a nutshell, through this VDJ rearrangement, they create different types of antibodies. This is the thing. And after pro B cells, base cells differentiate into pre B cell. This is an important maturation checkpoint. Why? At this pre-B stage, part of the receptor of B cell receptors is transiently expressed on the cell surface, and the B cell receptors finalize the gene arrangement. Okay, I'm not going into this. If I just tell you in a very nutshell that if this and the IgM is expressed on the cell surface, as you can see, as is expressed, but it is immature, not functional. Even then, it is expressing its on its surface the IgM. Then it is getting selected, and it will go to the spleen, where it will, if it is getting exposed to the proper stimulation, it will be a mature B cell producing thousands of antibodies. Okay, this much is good enough for you. Uh, Before cell is matured and leaving the bone marrow in the final stage, all B cell must be tested to make sure they do not recognize antigen of their own cell. This is very, very important. What is that? If these B cell recognizes own cell, means own body cell, as foreign, then autoimmune disease will start. So if it recognizes its own body cells, then those cells are get destroyed. Only positive positively selected cells are remain functional. Okay? Similar thing, you don't have to go into this. Similar thing happens with T cell. T cell also have the common lymphoid progenitor. Here also it is common lymphoid progenitor. That same cell, from there it produced B cell. Here it will be producing precursor T cells. Okay, so it can be CD4, or ultimately it will be CD4 positive or CD negative. Here it is not having any of these two, CD4 or CD8 positive. Okay, it must be having then what? Eventually it must be having CD3. Otherwise, it will not be able to recognize, we will not be able to recognize this cell as T cells. From here, it will go into the thymus. What will happen? If these cells, these precursor cells, they get in the, now where they are staying, where they are residing inside the thymus. Thymus will test whether these cells have the capacity to become a functional T cell or not. Suppose you are going for an examination for us, you want to be a soldier and you are going into the selection procedure. You want to be a soldier, but those people those people or the judge will judge you first whether you have that capacity to be a soldier or not. How they will? They will first check whether you have any integrity for your country or not. If you are and or you don't love your country, they will not recruit you or they will not let you be a soldier. Same thing happens here as well. They first check whether these cells can peptide or foreign peptides or not. If they can recognize foreign peptides presented by MHC molecules, one or two, then they will then they will be positively selected. What is written here? MHC molecule plus peptide, but it's not very tightly it is recognizing, moderately. If it is very tightly recognizing, then it will not be selected because that can lead to uh, self-destruction, I mean autoimmune disease. If it is not relate, it is the or the interaction is very weak, it will also be not selected. Moderate interaction of these cells with cells having MHC and peptides will be T cell or B cell. A T cell, CD positive, 4 or CD8 positive cells or CD4 positive cells. The same thing. This is just for interest. This is the last slide. What is what I am saying? A T cell for selection must be exposed to a cell which is having MHC1 or 2 with the peptide. From where, for selection process, from where body will get such cell? In thymus, the cells of thymus, they are whether the B T cells, precursor T cells, can be selected or not. Imagine you are always using a cell of thymus. Quite possible that it is not killing or destroying the thymus cells. That is why it is positively selected. It is possible after being selected, it can adversely affect lung cell or stomach cell or cells from other organs because it has been tested with only one cell, that is of thymus. But it doesn't happen. There is a possibility, but that doesn't happen. Why? Because there is, because of this AIRE. Okay, what is this? The role of AIRE in the thymus is to introduce expression of many proteins that are not typically expressed in thymic cell. Thymic cells means cells of the thymus, such as proteins characteristics of lungs. So because of this AIRE, uh, though they are inside the thymus, they can test the whether they are compatible for other organs of our body or not. If they fail, they will be uh, they will not be selected. Okay, so this is just for your information. This is how it happens. Mm, or the AIRE full form is probably autoimmune regulator. Okay, this is what I want to say about it in the basic idea of TB, T cells, and B cells. And if you have any question, so I think if you have any question, please feel free to ask. Okay.
Students, please use your hand signal so that I can call your names. Yes, there is a hand from Ruhi. Please go ahead, Ruhi.
Good evening, sir.
Good
Um,
evening.
Sir, in the last class on 11th, I guess,
Yeah.
sir, you said that the peristaltic movement, it is a form of innate immunity.
Yes.
Yes, sir. Like how? क्योंकि whenever we hear the thing, immunity, हमारे दिमाग में आता है defense. तो
अच्छा.
how
Then
how does like peristalsis, play a role in defense?
okay. See, in conventional way, huh, in conventionally, you cannot say that yes, totally peristalsis or retroperistalsis is a part of immune system. But you will find the peristalsis is a part of immune system in some literature or in some books. You will find peristalsis is a part of immune system. Why so? कभी हम सबको कभी ना कभी तो लूज मोशन हुआ है ना. घर में क्या कहा जाता है कि इसको लूज मोशन हुआ है तो जर्म्स बाहर जा रहा है. ठीक है, कहा जाता है ना. डू यू से दैट? Yes.
Yes, sir.
So
Yes, sir.
just imagine the way it is if something happens, it needs to be out. The way it needs to be removed, you need that peristalsis. Okay, one. So again, if you eat something toxic, if something is not good for your health, what happens? You feel you vomit. So that is retroperistalsis, and the body throws it out. So that is retroperistalsis, and the body throws it out. Generally, it's more an eating is something is irritating your bowel, so it is coming out. But hypothetically, it can be considered as a part of immune system. It's a very good question. Generally, retroperistalsis is not considered as a part of immune system, but in some literature you will find peristalsis as a part of immune system. But generally it is not. Most of the books, they don't consider it as a mechanical uh, barrier of immune system. Generally they don't consider it. Very good question, Louis.
I think to act, I think the same thing happens for coughing and sneezing
But
also, isn't it?
see, conventionally we consider coughing and sneezing as a part of
I think what happens for aflatoxin or any other kind of food poisoning, the best thing that the body can do right at that point is to flush out, as you have rightly mentioned, and to reduce the time it has to come in contact with the gut epithelium so that it can cause limited inflammation. That may be the case. And Ruhi, I think you have another question. Please go ahead.
Yes, sir.
please.
Sir, suppose if there is an alternation in the suppose if there is an alternation in the T cells, sir, what ill effects does it like
I'm
cause?
not getting your question, Louis. Uh,
Sir, in the T cells, if there is alternations or
alternation means what you are trying to say?
सर चेंज हो
कैसे
जाता
बताओ,
है जो
आप
उसका
बताओ कैसे, मतलब कौन सा चेंज के बारे में, कौन सा चेंज के बारे में बता रहे हो आप?
You mean to say the changes in the surface receptor? Is it?
यस सर यस
Okay.
सर
Okay.
Which one? Okay, surface
ये
receptor में. हाँ,
यस सर यस
बोलो.
सर सर
I
सरफेस
think
रिसेप्टर्स अगर चेंज हो जाता है ना
हाँ,
तो फिर उसका क्या इल इफेक्ट्स हो सकता है
I think she wants to ask, sir, that you discussed about many of the surface receptors with particular CD values. She wants to ask if there is any chance that due to certain, mutation or any other thing, is there any chance that these surface receptors may change, or either physically or in their activity of binding towards the target cell? And if so happens, those, ill-trained T cell, what would be the fate and what would be the causal effect that may come up with? I think this is a
okay.
question she wants to ask.
Anyways, Rui, good question, Rui. If I, hello, yeah. See, I have mentioned there are so many receptors. Okay, I have mentioned only a very few, just for mostly CD4, CD8, like this. Okay, so just imagine